Hi, Andreas. Thanks so much for joining me today. I appreciate it.
Hello, David. Thank you very much for having me. Thank you for the invitation.
Absolutely. Just to start off, how would you describe your main goal in the research you're doing on ovarian cancer, and how would you present that to someone who is new to this topic?
Yeah. So recently we had this project on ovarian cancer, and we were very interested in the early molecular events in ovarian cancer. To do this, we assembled a cohort of very early fallopian tube samples with dysplastic lesions that are thought to be the precursor lesions to ovarian cancer, all the way to invasive and metastatic ovarian cancer. On these samples, we applied spatial proteomics and transcriptomics that we integrated so that we get insights into the early molecular events, the proteins or transcript being upregulated or downregulated along the disease gradient or transition.
And there were a lot of different terms that I was hoping we could define today because some I was unfamiliar with. Transcriptomics is different from proteomics, and how do they each help us learn about cancer in general?
Transcriptomics and proteomics refer to the study of transcriptomes and proteomes, which are the totality of transcripts and proteins present at a given cell or sample at any time point. We use these methods in cancer research, among other types of research, so that we identify potential targets for drugs or biomarkers for prognosis and diagnosis.
My understanding is that you were using an approach that involves AI, and I'm referring to the Deep Visual Proteomics. So I was hoping you could discuss what else people should know about DVP in terms of how is AI really assisting this analysis?
So in our lab recently, like in the past 3 or 4 years, we have developed this new method. We call it Deep Visual Proteomics or DVP, which indeed uses deep learning to identify cells of interest, segment them, perhaps classify them in different classes based on the markers that they express or don't express, based on their cell size, shape, or proximity to other cells. We start with this deep learning to identify the cells of interest. Then using laser micro-dissection, we isolate these cells from the tissue. And then we proceed with sample preparation for mass spectrometry and mass spectrometry measurements. So we apply this method, which is a spatially resolved method, while we retain information about the cell types that we collected. We get proteomic information at a very deep level, so typically thousands of proteins per sample, even from very, very small samples. This differs from what we did maybe 5 or 10 years ago, when we would just have a whole tissue section of a cancer patient, for example, who would scrape off all the tissue and analyze the bulk proteome, so the proteome that came from all the cells in this tissue section. But there are problems with this approach because let's say you have a portion of cells overexpressing a protein and then another portion of the cells circulating the same protein. These opposite effects would cancel out. So you wouldn't be able to identify this protein as an interesting protein, but there is some pattern that you would like to identify. So having this method, Deep Visual Proteomics, for specific types of cells that you're interested in, let's say, for example, the regulatory T cells, you get a more pure proteome coming only from the cells you're interested in.
One thing that really stood out about your research is not only are you using some very cutting edge techniques and a lot of groundbreaking approaches, but obviously you have the super high end equipment that goes along with that. I found out about the EVOSEP company and platform at the American Society of Mass Spec conference the last few years. You also noted using automated mass spec sample prep and the Thermo Orbital Astral. I was hoping you could explain how all these different types of high-end equipment really fit in and why they're a good fit for your work.
Yeah, that's a good question. So in our lab, we're all for high throughput. So measuring as many samples as we can in a single day. We typically process the samples in 384 well plates. So you can imagine that if we were to pipette manually, it would be a lot of manual work. And I mean, even if you would have the multi-channel pipettes to do this pipetting, we typically work with very low volumes, let's say 1 to 10 microliters. So you can imagine that you have your 384 well plate, you start pipetting. By the time you pipette the last row, the first row is probably already evaporated. So we use liquid handling systems that do all of that for us. So they're very fast, very precise, accurate, and also, we can be a bit more relieved or sure that we're not making mistakes because of all the samples we're pipetting. So this is the automated sample preparation. Then because we have this throughput to prepare many samples per day, we also want to be able to measure these samples very fast. That's why we use the liquid chromatography system from EVOSEP, which is a very robust system that offers relatively short gradients while maintaining still very good proteomic depth or coverage. And then to be very sensitive and identify many protein groups, we use the Thermo Orbital Astral system, which was the latest and greatest, allowing very deep proteomic characterization.
For my work, my company manufactures evaporation equipment for our sample prep. We're more working with the 96 well plates because after an extraction, you have a need to evaporate off that excess methanol or to concentrate the analyte because you have too much of that solvent. But with 384 well plates, much smaller volumes and that concentration, as you mentioned, sometimes it's already evaporated.
Exactly. That's one of the reasons why we go for the 384. Other reason is that you can also go for very small volumes, which is beneficial when you have very little protein in your sample coming from very few samples or even a single cell. The amount of exposure or contact that your protein will have to the plastic of the plate, even though we're using low bind plates, it will still play a role and make a difference. So the smaller volume you have, the less of the already tiny amount of protein you have will be bound to the plastic of your plate.
That makes sense. As we noted, you're using a lot of advanced techniques, but how are you able to gain new perspectives compared to if you relied on approaches that were more the traditional ways to research cancer?
So in a traditional kind of cancer research, you typically, have one target, one protein, for example, that you do research maybe for your whole PhD, 4, 5, or even 6 years, just on this same protein. By applying transcriptomics or proteomics, you can have multiple targets at the same time. My professor likes to say that every time you do a proteomics experiment, it's like you do 10,000 Western blots at the same time.
That's a great explanation, and that's something that I think the audience will be able to connect in terms of their understanding with known techniques to some of the more cutting edge technologies. Could you introduce ID01 and the paradox that you discussed in the research to the broader audience?
All right, yeah. So IDO1, or indoleamine deoxygenase, is a metabolic enzyme. It converts tryptophan to kynurenine, so catabolizes tryptophan. And it is known that tryptophan is very important for immune cell function. It is also known that in some cancers, among them also ovarian cancer, IDO1 levels are elevated, which means that you will have depletion of tryptophan from the tumor microenvironment, which then leads to T cell anergy. So elevated IDO1 might be used from cancer cells to evade our immune system. That's why it was thought that using IDO1 inhibition would be beneficial for cancer patients. But in practice, in the clinical trials, the IDO1 inhibitors failed. That proved that we don't really know everything about IDO1 biology. And in our study, we identified a new role for IDO1 that is exactly the opposite from what we thought initially. So we saw in our study that IDO1 is very important for cancer cell death. So by inhibiting IDO1, you protect cancer cells from death. So this might explain why IDO1 inhibitors didn't work in the clinical practice.
Your studies found several new proteins and pathways as potential therapy targets. What would you say are some of the potential real world impacts for patients? And what steps are really needed to translate some of the findings from this work into actual clinical treatments?
So we're a core proteomics lab. So we mostly do the initial steps of drug discovery, which is the target identification, the discovery. So we identify these targets that you mentioned. And then we even went a step further and we did some functional validation of these targets, some of them in vitro, one of them even in vivo in mice. But this is not the same as saying that tomorrow we will have some new drugs for ovarian cancer, because of course one would need to do many more experiments still. For example, animal experiments, mice, rats, and so on. And then if the results of these experiments were very promising, then go the clinical trial path and then clinical phase 1, 2, 3. If the drugs look safe and effective, then only then you can have the clinical application and really save lives with these drugs. So we're still many years and many hundreds of millions apart.
For academic researchers or students who are just starting in their chemistry journey, what are some of the fundamental skills or um, experiences that they should be working to gain to really enter this field and have an impact on it?
I'm not sure if I have anything specific for cancer researchers, maybe something in general for researchers. Research is very disappointing and frustrating oftentimes because, you do all these experiments many times, or maybe most of the times they do not work. This is not necessarily because you did something wrong. It might very well be that it is just the nature of the experiment you're trying. So I would say one needs to be resilient, not get frustrated too easily, just persist and insist, and maybe learn from the mistakes.
I think that's a great answer and something worth acknowledging in terms of that. Sometimes there
Exactly.
will be
Just keep
that
pushing.
disappointment.
It can be frustrating, but the one time that it will work, it will be so exciting that it makes it all worth it.
Absolutely. And yeah, I'd love to hear about that too, in terms of some of the more exciting findings that you've made, if anything sticks out, some of those major wins.
Well, I would probably stick to the IDO1 paradox that we just mentioned, because that was really not expected. So in biology, and in general in science, in most of the cases, things are not black or white. The understanding that we had back then when we were trying the IDO1 inhibitors in the clinics was that IDO1 is bad, so you want to inhibit it so that you get benefit from the patients. And then we were doing experiments and we saw something that didn't make sense, that was against the common understanding. And of course, you can be wrong. So you need to check again and make sure that your experiments are correct. But then once you have it consistently in your experiments and you see something that you didn't expect, maybe there is the next clinical breakthrough over there. So just have the open mind to accept something that you didn't expect and try to explain it.
Absolutely. Yeah, I think that's some great insight and good advice for anyone who's just starting their academic and research journey. Andres, what else about your research would you like people to know that we haven't covered?
So we spoke mainly about ovarian cancer. This was a big part of my PhD. I'm a PhD student now approaching the end of my PhD after 4 years. So I should be defending in a couple of months or something. Ovarian cancer was indeed a big part of it, but I was very interested into the methodology, this deep visual proteomics that we mentioned at the beginning. And I tried to explore many different applications of this method in different types of diseases. So besides ovarian cancer, I worked on lung cancer, skin cancer, osteoarthritis, muscle biology. So maybe the message here is that we have developed this spatial proteomics method that is applicable to many, many different types of questions and diseases. We're trying to demonstrate the applications of this method so that more colleagues out there apply it in different topics, in different questions, so that we have a greater clinical benefit at the end.
Yeah, it's certainly good to hear from someone who's done so much and getting ready to defend, and I'm very happy for you and all the progress you've made. I really
Thank
enjoyed
you.
learning about your work. And
Thank you very
um,
much.
what do the next steps look like in terms of your work? Do you see continuing to focus on this area, or do you see a pivot after this defense?
That's a good question. And I have not figured out the answer to that yet because, I mean, maybe in 2 or 3 months I'm defending. So I should be looking for jobs right now. But I have been busy with all these publications and now writing my thesis that I didn't really
Mhm.
take the time to think of the next steps. So, yeah, once I'm done with my defense, maybe I will start thinking what the next steps are.
You can only handle so much at once, right?
Exactly.
Andres, I really appreciate the time today, and I look forward to sharing some of this groundbreaking research with the listeners of the podcast. So thanks so much.
Of course. Thank you so much, David.